So my understanding that this group is interested in both AI and aging. So my lab is 50% computational, 50% experimental, and that's basically what we try to do. We try to solve the problem of aging. That's basically in essence what this is. So I will tell you a little bit about how we do this by quantifying aging, longevity, and rejuvenation, because to me it has been this kind of advance has been a major development in the field. So first, when we say aging, we need to ask what is aging. And so you already heard the insights from David Sinclair. So I think it's very, I actually enjoyed his talk. They actually work in the same building on campus, so interestingly, and also collaborate. But I will give you this example because that's actually a very difficult question to answer. So 2 years ago, I chaired a conference called Systems Aging, Gordon Research Conference. So all experts in the field, about 200 people. And we gave a questionnaire to these people asking the very basic questions. Like what is aging, what causes aging, what is rejuvenation, is aging a disease? 10 questions like that. And the answers, over 100 answers, there was not a single point of consensus for any of the questions. And the definitions of aging, basically 100 responses, there were 100 definitions of aging. I tried to cluster them, and this is what basically it is. So that's not how they answer it, but it's just a kind of summary of how people think in general. These are experts in the field. So some people think of aging as the increased chance of dying. Like you talk to many demographers, and all of the Gompertz equation and most of the assays in the field are based on mortality. That's how we study aging. For some people, it's mortality rate. Other people say no, it's loss of function. With age, we decrease VO2 max or muscle strength or vision. That's aging. Other people say no, it's just a process of continuous development. We develop, and there's a certain trajectory, and this trajectory continues, and that's aging. Other people say no, it's biological age increase, so damage accumulation, or whatever, many other kind of features. In the end, we use word aging to designate different processes. You ask, what do you study? Aging. What do you study? Aging. But maybe you study completely different things. Of course, many of these are must be manifestations of the aging process, not aging itself. And they relate to one another, especially late in life. But in order to target aging, we need to understand it, right? We need to find the essence, not just formal definition, but just basically the essence of what is this. And that's incredibly difficult. So quite a bit difficult. So in another way to kind of look at this, you can say, well, aging may be mortality. But to me, we die because of diseases, right? So you could look at instead aging as the incidence of diseases like cancer, diabetes, Alzheimer's, heart disease. But diseases are caused by functional decline. And functional decline is caused by accumulation of deleterious changes such as molecular damage. So to me, I've been thinking more in terms of damage, on the damage side. So my view on aging is based on damage, basically. And I will tell you a little bit in more detail of this. I was originally trained as an enzymologist, biochemist basically. And we work with enzymes, at least used to work. And so if we consider an enzyme, enzyme converts a substrate to a product. So what can we say about this enzyme? This enzyme has a gene, encoded by a gene, and there is a purpose of this enzyme to convert a substrate to a product. That's the reason we have that gene. We have many genes that code enzymes that do this particular function. But if you look at the enzyme is a molecule composed of 20 types of amino acids. Has a particular structure, and no matter how well the active site is built, it's not perfect reaction. It cannot catalyze the reaction perfectly. Actually, in chemistry, nothing is perfect in chemistry. Therefore, nothing is perfect in biology too. Therefore, there's non-zero likelihood this enzyme will produce something else. Let's call it damage. It could be a byproduct of mutation or epimutation, whatever. A change, altered expression of a gene, damage. So now, product, conventional product, is a function, right? It has a program. We can call it a program. Yeah? Damage is not a program. There's no purpose in making damage. It just happens because we have this blue process, right? But it's not just any damage. This damage is produced by this particular enzyme. Therefore, this feature has this programmatic nature. So the blue is programmed, red is programmatic, but not programmed. And to me, this red is damage, and therefore is aging. In other words, aging is not programmed. It cannot be programmed. There's no purpose in aging. There's no benefit in aging. So, on the other hand, genes, they're programmed. They have a particular function. Development, it's a program. We could disrupt development, like disrupt the gene, we stop development. We disrupt the gene for apoptosis, we stop apoptosis. We cannot disrupt aging by manipulating a gene. Although it's very clear that we age very similarly, they have programmatic nature, but it's not programmed. Yeah, that's very important. Now, we can extend this concept and say that this damage can be represented by byproducts, mutations, epimutations, imbalance of protein complex, any other known and unknown forms of damage. We call it deleteriome often. It's kind of similar to cumulative damage, but the deleteriome is not just damage, because it's any negative consequence of being alive. In other words, aging happens because we are alive. We live, we have processes that do whatever they do, and they produce negative consequences as a result. And these negative consequences accumulate. That, to me, is aging. So the important point is that every cellular function contributes to damage, and therefore to aging. There's no major damage form. There's no point studying DNA damage repair or like mitochondria, autophagy, or protein synthesis. Aging is a systemic process. The whole system lives, and therefore the whole system ages. That's why it's so difficult to describe it, quantify it, and target it. So far, nobody succeeded in it, I would say. Oh, another point is antagonistic pleiotropy. Who knows what antagonistic pleiotropy is? Can you raise hands? Okay. Okay. This is maybe an evolutionary concept which is the most important in the aging field. Basically, I'll tell you, it was Williams proposed it about 60 years ago, and that's basically the dominant theory currently in the aging field, called antagonistic pleiotropy. Basically, the idea is this: you have a non-aging organism, yeah? And then you evolve a gene variant, or maybe gene, that makes an organism, when it's kind of in the younger state, better functionally. It may jump higher, or run faster, or be smarter, or see better. So, of course, it will be selected, will be preserved, that variant, even if that variant will produce some negative consequences later in life. Right? You cannot avoid it, it will be selected because it's beneficial early in life when organisms reproduce. And therefore, Williams suggested it's unavoidable that aging evolves. Unavoidable because these such variants give benefit. These organisms will outcompete. But what I'm saying here is this model, it's not correct. Because if you look at this scheme, that's exactly antagonistic pleiotropy. Basically, every function already has positive, like blue, and negative, red, negative features. Therefore, every biological function has antagonistic pleiotropy features. Therefore, antagonistic pleiotropy cannot evolve. It's there from the very start. So, of course, it can be used to study changes in lifespan, whether organisms age faster or slower, but it cannot explain evolution of aging, conversion from the non-aging to aging state. Like bacteria which don't age and symmetrically divide, they have antagonistic pleiotropy features because they have this any chemical reactions or catalyzed reactions. And so basically this is my understanding of the aging process, and with this understanding we built a research program around this. And I will tell you how we try to use it to address questions in the field. First, the basically lab operates like that. We study longevity, and here we study basically the potential of the system to live long. It's not actually change, it's that when we change longevity, we change the potential. We like convert a short-lived mouse to a long-lived human. Yeah? How can we convert like a short-lived organism to a longer-lived organism? And we study at the level of cell types. Some cells are short-lived, like cells in the blood sometimes live 2 days. And cells in the brain, like neurons, they live whole life, right? They have same genome, same kind of organization, but why the different lifespan? Longevity across mammals. Mammals have a similar genome size. They evolved from the same ancestor. There's 200-fold difference in lifespan. Like some shrews, they live 1 year, some whales can live 200 years. So nature apparently can easily change lifespan by manipulating genes and go from like 1 year to 200 years. How does it do it? We try to understand that. And then also longevity across interventions. Because we now know many interventions and many species, we know like 15 ways to extend lifespan in mice, or whatever, in C. elegans, flies, and so on. And this results in, we call it signatures of longevity. These are omics, typically omics patterns, that indicate the potential of the system to live long. We could target these patterns, and therefore the system would convert it from a shorter life to a longer life. But here we study also aging. Aging is age of the change. And this can be quantified by, I'll show you some clocks, like epigenetic clocks, atomic clocks, organ-specific clocks, and so on. It results in actionable biomarkers of aging. And the third component is rejuvenation. Rejuvenation is, to me, is the conversion of the older state to a younger state. So when we look at the aging, I already told you what aging in my mind is. And if you kind of think, if you accept that model, the aging would be irreversible. Because there's a myriad of different processes and modifications and byproducts and damage types which cannot really go back. You cannot just go in the reverse way. But you could still target biological age and to reduce it. And I'll also give you some examples. Therefore, it's also possible to rejuvenate the organism even though aging is irreversible. And all the way it allows us to target aging and find interventions. And we have many projects and we identified many interventions that are known to extend lifespan. We have a huge transformative project from NIH where we actually have a large scale of mouse studies where we in silico predict new compounds that extend lifespan and test them in mice for lifespan and other features. We already identified many, although it's still unpublished. So now I will give you one example, or just one example of the longevity study. And this is from a paper published in Cell last year. So this is experiment correlation matrix of three kind of features. Blue, longevity; red, aging; green, interventions that extend lifespan. Blue, see, brain, like kidney, liver, these are changes associated with longevity across mammals. And you see they correlate with one another. If something changes in the brain, typically similar things change in the liver or kidney. Red is aging, like aging muscle means muscles of different mammals. Brain of different mammals. Human means different tissues of human. Global means different tissues and different species. Yeah? They all correlate with one another. And this is interventions. This is like a calorie restriction, rapamycin, acantharyflosin, icarbose, growth hormone deficiency, and so on. They also correlate with one another. But when we relate these three features of longevity and aging, it's very, to us was really puzzling, actually, very striking observation. Most striking here is that we compare this corner kind of with that corner. There's no correlation of longevity across species with longevity across interventions. Whatever we study in the lab, as a field, calorie restriction, rapamycin, whatever genetic manipulations, lifestyle changes, there's no correlation with how nature extends lifespan. It's really amazing because the common paradigm, let's go to the whale, find, okay, why does it live long? Find that gene, express in the mouse, and make mouse longer live. And typically, it make mouse longer live, but maybe 5%, 10%, not 200-fold. Right? So there's no correlation. And to me, this represents really an untapped potential of that longevity across mammals. So to me, kind of explanation is this. So nature changes many pathways, many genes. It's not a single pathway, single gene that we typically target. And therefore, mechanistically, it's also becoming. But the point is that all of this, they provide us opportunity to find new interventions, different types. Type of the longevity across mammals, one type of interventions, and it can be. Anyway, so this is about longevity. Now about aging. And in our field, about 10 years ago, a revolution happened. This was started by Steve Horvath, who used to be at UCLA, now Daltos Labs, who in 2013 published a paper on the first multi-tissue human epigenetic clock. Basically, it's a tool that predicts biological age of an organism. And we followed closely, and we developed the first mouse clock. So I'll just give you one example of our study, but it's kind of conceptually similar. So this is the actual age of mice, this is predicted age. And this is the 16 age groups. You see, there is good correspondence. How we do it, we take mouse blood, we sequence it by this method called reduced representation bisulfite sequencing, and apply various machine learning approaches to identify a set of CpG sites. Do you guys know CpG? Oh, sorry. Okay, so DNA, 4 nucleotides, one of them is C, cytosine. It can be methylated or not. And we follow methylation changes with age. Yeah? So some cytosines increase methylation with age, some decrease methylation. Out of the millions of CpG sites, we find a subset that best represent the aging process. And we have a model, each site has a weight in that model. Together it's like a mathematical formula that predicts the age. And this is how it works. And if we subject mice to calorie restriction, like 4 age groups here, you see they are all biologically young. So in other words, we can quantify easily the effect of interventions that are already known to extend lifespan. Well, this is an example. We take adult cells from 10-month-old mice and reprogram them to embryonic cells called iPSCs, induced pluripotent stem cells. This is the discovery of Yamanaka, I think the most important discovery of this century. He was given the Nobel Prize very quickly for it. And he converted to adult cells to embryonic cells. But for us, it's a rejuvenation process. We find that these cells are predicted to be of 0 biological age based on the clock that we have in the blood. So it does work. So, but the question is, like I showed you that we train the model based on chronological age. But in fact, we don't know what's the best way to model. We could model based on chronological age, like here. We could model based on mortality, like time to death. We could model on age-related diseases, or functional decline, or damage accumulation, or other features, like rate of change, for example. And all these biomarkers would quantify aging just a different way. What is the best way? Actually, it's unclear. So, kind of these features represent the best known clocks, like we have Horvath clock trained for chronological age, we have like a Grimm age clock that trained more on mortality, we have Tunisian Bayes clock that is trained for rate of change, and so on. So for this, we formed a consortium called Biomarker of Aging Consortium. This was really led by Madi Mukri, who's an instructor in the lab. And this is the first paper we published, and it's been really successful. About 30 leading labs in the field joined efforts to, and actually this consortium provides a platform for debate and discussion and development of new biomarkers. It's been quite successful, so this website, I want to mention this conference that we'll have next week. So of course, you're welcome to join.
And by the way, for a certain category of winner, you will get a free ticket to the Aging Consortium conference next week, so do good.
Yeah, thank you. So yeah, last year we had it at the Bach Institute in California. There was 250 people showed up. We couldn't accept more because the room was limiting. This year we have a room at Harvard Medical School which holds 500 people, so we expect a full room as well. And we have representatives of governmental organizations, editors, industry, like big pharma, whatever, X Prize, Evolution, so on, RPH, new agency, they all will be there. So we also have a challenge to predict, to compare biomarkers, aging biomarkers. And just yesterday, I think there was a piece in Science about our challenge. Just they kind of described it, and actually you're the first to see the results. They will be shown at the conference. But basically, these are the existing plots. What we did, we took 500 people from our hospital, MGH, MGB, Mass General Brigham. 500 people, we profiled them for DNA methylation. We know their ages, we know who they are, we know how many of them died. And we just ask people, okay, tell us, what's the ages? What's their ages? How long do they live? They live because they give blood, and then they lived a certain time and then died, some of them. And these are the 3 winners. The actual age, this is from about 20 to 100 years old. And this is the predicted age. You see, these biomarkers work really, really very nicely. This is, we had over 500 submissions to the 37 teams qualifying to the final. And you can see this at the top, this mean absolute error. All of the top scores are better than existing biomarkers. Really amazing. Like just maybe like 2.5 years or so mean absolute error. And these are some people from the industry, some people just kind of graduate students. So I invite you all to compete as well. So it's a first phase. Now we will announce next week results of the second phase, the prediction of mortality. This was prediction of chronological age. So we don't know yet, but we are finalizing the results, and so we will announce at the conference the winners, and the winners actually also will give short talks at the meeting. Okay, it's a short presentation. I just want to quickly mention a couple of studies from our lab recently, which I will not discuss, just mention briefly. So Albert King, who's a very talented graduate student, he developed the first causal clock, which was published recently in Nature Aging, because the problem with the current biomarkers, and there are many issues, right? But one issue is the causality. So I'll give you one example. You can predict the age of a person by features like wearing glasses, using hearing aid, kind of quantifying number of visits to doctors, using wheelchair, walking cane, and like take 100 parameters like that, you can predict roughly the age of a person. Okay? And then you fire all doctors, like no doctors, like nobody can wear glasses because nobody can, doctors cannot prescribe it, right? Based on this example, everybody is young because they don't go to the doctor's office, yeah? But of course it's not. Same way at the biological level, changes, there is a damaging changes, like damage happens during aging, but organism responds to damage. We call them adaptive processes. And these adaptive processes also have age-related structure, and therefore they end up in the clock. So the clock in biomarker is a mixture of different processes, neutral, damaging, adaptive, but they can quantify age, but we need to use biomarker to also test interventions. And when we test interventions, it's completely different. So our intervention is to fire all doctors. Yeah, it's wrong for that biomarker, right? So he developed his first causal clock. And this clock works specifically with interventions that extend lifespan. And Andrei, who's a physicist, theoretical physicist, he was able to identify stochastic and regulatory components of aging. Also really kind of amazing work, and this was done by comparing in single cell and bulk kind of data. Let me give you some examples of how we study. This work from Jesse Pagani, constructed in the lab. He found that biological age can reversibly change by severe stress. Like basically under condition of stress, and these biomarkers, like for example, epigenetic clocks, like Grimm age, pheno age, and so on, increases, and post-stress it goes down. And we described 4 cases, like for example, major surgery, severe COVID-19, pregnancy, and parabiosis. Like for example, pregnancy. We find that women in the 3rd trimester, they are biologically older, and postpartum, their biological age is reduced again to normal. Or for example, COVID-19, we find that the maximal biological age for people who get to the hospital, to the ICU, to the ventilator, at that point the biological age is the highest. Once they recover, it goes back to normal. So we currently expand this because we have other models like sleep deprivation we study. We even now study how biological age changes during the day. That's actually a very difficult question. We're trying to do it at least. So we also developed, this is also a paper, it's unpublished, but it's under review in Nature now. Really amazing transcriptomic clock. Alex Tishkovsky, who is an instructor in the lab. It's 4,500 mouse samples here, RNA-seq, so it's kind of a huge data set with all kind of manipulations that change, like accelerated, decelerated aging, genetically, pharmacologically, and so on. This is how it works, chronological, actual age, predicted age, and this is how it works with mortality. We developed a clock based on future mortality in mice, actual data and then predicted data. And this is application to interventions, control, this is lifespan extending interventions. You see it works, and then lifespan shortening interventions. Works in both ways. So it can be used to identify new interventions. In diseases, it shows that this transcriptomic clock that in each case of every disease has a higher biological age than controls. So apparently when we have a diseased tissue, it literally becomes older than other tissues. Let me give you two quick examples of rejuvenation. This was done by Bohan Zain, who at that time he was a graduate student, now stays as a postdoc. He used the model of parabiosis. We take a mouse, old mouse and young mouse, we sew them together so they have joint circulation, they exchange blood, and keep them for 3 months. After 3 months, we separate them, wait for another 2 months, and then wait until they die. We basically find that after 3 months, old mouse is rejuvenated. It dramatically decreases biological age. We wait 2 months, it's still, the effect remains, still younger, and then eventually it lives longer as well than control. So this is actually one of the most robust examples of rejuvenation. We can rejuvenate animals, but in this case, of course, very extreme, when we connect mice like that. Another example of rejuvenation shown here. This was done by, well, first was conceptual paper, but then experimentally was done by Csaba Kiribeshvili and Alex Csaba. Csaba is now has his own group in Hungary, and Alex he leads bioinformatics at a company called Retro. We found the first natural example of rejuvenation. So when life begins, here we have a zygote. Zygote is a fertilized egg. Yeah? We have a egg and fertilized with sperm, that's called zygote. But interestingly, the biological age of the zygote is not 0. It's low, but not 0. And then initially it stays the same and then drops here. We call it ground zero state. And that's where we think aging begins. When you ask a question like various researchers in the field, when does aging begin? Most people say after completion of development, or maybe when at the age of puberty, or the age of numinal mortality, or when the organism is born. We find that aging begins here. But in humans, about 3 weeks post-fertilization, post-conception. So we actually think that actually the organismal life also begins here. So this phase is a phase of kind of preparation for organismal life. When you have a cell, it just divides, called cleavage, into multiple cells. It kind of extends telomeres, clears damage, prepares in other ways, also decreases methylation, remethylates, and that's when it's ready to go, it goes. Like it makes a germline, organogenesis begins, it's all here. Yeah, that's when aging begins. But we think so. I would say it's still a hypothesis because it has so far has only been demonstrated in our lab. We really wait for somebody to validate it. We publish this in mice, at the bulk clock, single cell clock, in humans. There's a paper by archiving on frogs, but it's possible that we're making some mistake here. I don't know yet, but I think would be interesting to test. The main point is that this is the first natural example of rejuvenation, and we don't know mechanistically how it happens.
How do you know that it happens?
What?
How do you know that it happens?
Because we quantify with our biomarkers of aging.
Okay.
So we used like bulk epigenetic clocks, like a Horvath clock, like image and so on. And then Alex developed a single cell DNA methylation clock. It's a probabilistic clock, completely different. This was Nature Aging paper. So also found the same. And when we apply now transcriptomic clock, also shows the same. In each case, also the timing is the same. And also, just conceptually, it makes sense because the germline, it ages an organism. Like a sperm from an older man is older from a younger man, right? Oocytes from the older woman are older from the young woman. And then when your zygote is formed, the age does not disappear, right? This damage is there. So conceptually, I was thinking it must be rejuvenated. It must happen. It's just for some reason, I don't know why, nobody paid attention to it. Like everybody assumed zygote is formed, life begins, that means it's just a 0. But I don't understand, like you have the most differentiated cells, like sperm and oocyte, the most differentiated. Now they fuse, suddenly everybody calls it the least differentiated zygote. It makes no sense. So if you look at all these books, developmental biology, I think it's all wrong. At least at that point. So I think this stage of de-differentiation, you prepare for organismal life, and kind of organismal life and aging, they begin at the same time.
What is this name of this stage, which is in the bottom?
Gastrulation.
The stage.
Yeah. I have one more topic. If
Go
you have
for it.
time.
Yeah, totally, go for it, yeah. I'm
Okay.
enjoying this a lot.
Okay, so one topic. We have paper just got accepted. We're going to share with you. We always discuss, like, we tell, okay, how old are you? Whatever, 40 or 20. Okay, but we always tell like a biological age of an organism. But in fact, organism is composed of different parts. And there are different classes, potentially in different parts of the organism, right? So, and last December, it was a really amazing paper from Tony Scarei, he is at Stanford, who developed first organ-specific biomarkers. Technically, it wasn't that great because they didn't work that well, but conceptually it was really amazing advance, really one of the more important papers. But we had access to better data sets. We use UK Biobank, which is an amazing data set with 53,000 proteomic profiles in plasma. This is work done by Ludger Bimini, who is a postdoc in the lab. This is the clock that he developed, chronological age versus predicted age, based on the plasma proteome profile. Like, we have plasma in the blood. Plasma is like part of the blood. You have cells and plasma in the blood, yeah? And this is a clock based on future mortality also, because many people died in that cohort. And then, what we did, we find, okay, what is plasma? Plasma is composed of proteins and metabolites. They come from different parts of the body. Like maybe we find 100 proteins in the plasma, they come from the brain. They are made in the brain, secreted and found in the plasma. If we build biomarker based on these 100 proteins, we can predict the age of the brain. In the same way, like another 100 proteins from the liver, we can predict the age of the liver. So we profile plasma, and we can tell the age of different parts of the body, right? And so we have these clocks now. Liver clock, brain clock, artery clock, whatever, intestinal clock, kidney, and so on. Yeah? These are proteins that come like from the lung. In the plasma, we profile them, we tell how old is the lung. And then we can tell in the same organism what is the age of different organs. This way. Yeah? And these are residuals show, this is chronological model, mortality model. These are residuals. You can see there is strong correlation. This means like if somebody is old in the brain, also old like in the artery, you know, in the liver, in the lung. It makes sense, right? Chronologically and also in terms of mortality. Sometimes like things like artery and local age is like an age that we predict based on locomotor activities also. There is another way to predict it. But in general, yes, kind of good correspondence. Now, when we use these clocks, we find it amazingly we can study diseases. For example, we can use this clock, lung clock. Proteins go from the lung to plasma. We profile, we tell the age of the lung, right? We say which diseases are predicted. COPD. COPD is a lung disease. So lung clock best predicts COPD. We take liver clock, the first disease is liver cirrhosis. If you take like brain clock, we predict dementia. So really amazing. And then we have this clustering of different clocks and diseases. Like COPD, like people with COPD, with lung disease, this is the ages in the organs. The oldest is lung. Dementia, people with dementia, oldest in the brain, and so on. But also there's a cluster of like metabolic diseases in different diseases and different organs. So this is really have many practical applications. For example, hope nobody smokes here. I don't know, but nobody smokes here.
I don't.
Smoking is bad. So, but which organs are affected? Because now we have different organs, and we look for people who smoke. It's bad for all organs, right? But the worst is for the lung. Not surprisingly. But of course we see negative, there's no positive effect. Smoking doesn't show any positive effect for any organ, only negative effect.
What's conventional?
What?
What's conventional?
It's a conventional clock. It's combining them all.
Okay.
Yeah. So, how about drinking? Does anybody drink wine? Vodka?
Okay.
Nobody? Okay. So, it's more complicated. So, we find, okay, what are the organs that affect us? Like we find kidney, for example, intestinal aging is particularly worse. Yeah. But then we find there is a positive effect, like in the lung and artery. So interesting. So another way to look at this is like this. Just another example. Let's look at associations of foods with arterial age, just artery. Artery means proteins made in the artery go to the plasma, we profile them, we tell age of the artery, right? Wine is good, yogurt is bad. Yeah. If somebody has accelerated aging in the artery, they may benefit by drinking wine, and they may benefit by not eating yogurt. Yeah. But if you look the same foods, only intestinal aging model, it's the same people, right? Only now we have artery versus intestinal. It's the opposite. Wine is bad, yogurt is good. So what we think is that we should stop, not stop, but we should think, take into consideration looking for interventions that work in a personalized way. We need to know who can benefit from intervention because we all age in different ways. We can quantify it and we can tell, we cannot tell, but in the future we should be able to tell who can benefit from which intervention. Yeah? Like calorie restriction, it's known it's really helpful, but it will not benefit everybody. We need to know who can benefit based on their aging patterns. In other words, with asynchrony organ aging, we may target these weak links, organs with accelerated aging within the body, by developing interventions. So our model is like this. Zygote, remember I told you zygote, that's fertilized egg, aging begins here, grind 0, 3 weeks in humans. And then organs, systems, they age, but then some systems, they age more. But this is not simply an age state, because they age non-autonomously, right? They interact with other organs. So in a way, their age is decreased, pulled back by other systems. So it's not just older, but it's also altered. And we don't know yet how to describe that state. This one is, it's our model still. And often in the field, there's a question, like, is aging a disease? That's actually, there's like 100 papers just with that title. And there's always like a movement to classify aging as disease so that can be targeted. We actually had a paper now already 8 years ago saying that the question is wrong. We cannot really say. We cannot say, we basically said aging is not a disease and not a disease. But then we can say, we can reverse the question now and ask the question, is chronic disease a manifestation of aging? And based on what I told you, we suggest basically a new model that links together aging and diseases, but basically saying that disease may be viewed as a systems or subsystem aging, or maybe like pockets of non-autonomous aging. In other words, we say there are no diseases. It's just the different parts of the organism, like systems, subsystems. It could be at different scales, could be organ, could be individual cell, could be one gene. They age, and that aged, altered state is we call a disease. So I think it's a way to bring together aging and diseases, and maybe in the future this could help to target them. That's basically all I want to tell you. I will quickly say, just summarize so you remember what we discussed. We discussed this longevity signatures across species, cells, and interventions. These are basically omics patterns that indicate the potential of the system to live long or short. And there is amazing difference between species longevity and known interventions. We also discussed biomarkers a lot, at the bulk, single cell level, causal, tissue, multi-tissue, also at different omics modalities like genetic, transcriptomic, proteomic. I told you about reversible changes in biological age, a few examples, and also I told you about biomarker of aging consortium, and also challenge. We also discussed rejuvenation. I showed you three examples actually. Cellular programming I mentioned, heterochromatic parabiosis, and I also mentioned this ground zero model of rejuvenation. And finally, organ-specific aging. There's organ models that can be trained for chronological age or mortality, and also association with diseases of diets and new ideas about whether the relationship between aging and disease. But altogether, all we do is try to understand and target aging, longevity, and rejuvenation. So this is a very difficult problem. It's like I feel like we are still like at the very beginning, because like when you start studying aging, almost there's no barrier. Like we all see aging every day, right? So you start and you feel you already understand. You become like an expert. But the more you study, the more difficult it becomes. Like even with biomarkers, it's very clear. Like, okay, we have initial biomarkers. The more we study, the more problems. The elasticity, causality, cost, adaptation, this kind of different scales that we have, all these challenges that still need to be solved. And we need people to do it.
That's what I'm hoping comes out of this.
Yes. So this is the current lab. Basically, in the lab members, the former lab members, and collaborations and funding sources.
Oh, I love the logo.
Yeah.
That's kind of cool.
You go back in age.
Yep.
Yeah.
Thanks. That's it.
Do you have time for questions?
Yeah.
First of all, let's give it up. Thank you so much for making out on short notice. I know Elena was very kind to share what we were doing today. I do want to let other people ask questions because I'm sure they're out, but I had one very specific thing to ask you, if you have a moment. I've been very interested in sort of the way computational systems perceive and measure time from an info-theoretic perspective, right? I mean, so if you think about the brain, for instance, there are certain substances you can give the brain where it's going to perceive time a little slower or faster, right? So, if you take caffeine or if you're sleep deprived, all these different things can sort of change, your experience of the compression of time. And I've often, for a very, very long time, pun intended, wondered, is there an analog of that for the body? And I wonder for the rest of the body and other organs. And I wonder if we were to measure in the brain that if the brain was perceiving time slower, was it aging at a different rate? And would that then be applicable to the rest of the body and other organs? Because I also know, like in a lot of, longevity science, it's sort of this idea of tricking the body into a certain state, right? We have fasting, mimetics like rapamycin and all that. And I'm wondering, has there been any research into this sort of experimental idea? Just picking your brain.
That's a difficult question because it's difficult to define time.
Yeah.
Right?
Yeah.
I know yesterday Stephen Walker was
Yes.
here, and I was reading, a couple of weeks ago, I was reading his paper that was on time, right?
Mm-hmm.
What is time? It's an incredibly difficult concept. Time also, how time is perceived. I'm not an expert here to answer this, just because in my lab, I try to bring physicists, mathematicians that come and help. We also have one theoretical physicist, for example. So we discuss and we publish a few physical papers, but I don't know. The answer basically no. I may say I don't know too many of your questions because that would be honest answer, right? So we don't know. We have models, right? We can test these models, but we cannot explain everything.
Yeah. Any other questions, anybody? First.
My question is on these slides related to, let's say, the effects of smoking or the effects of certain foods. I believe that the units there were something like this increases your age by maybe 0.07 or 0.0004. Is there a way to understand that more intuitively, like maybe it ages you by your lungs by 10 years or brings you 30% closer to cancer?
But potentially it can be converted, but we would just need to do it. We haven't done it.
Okay.
So I mean, this is the first. The data set became available less than a year ago, so it was really kind of new study. Currently, we are expanding on it because there can be many phenotypes can be studied. But we initially wanted to do it just conceptual way, and actually I should say even that the examples that I presented, it really should be viewed as conceptual examples. We cannot be for sure, we don't know for sure like the effect of the alcohol, for example, or smoking. These are associations. There's it's for presentation purposes, I would say, more or less. But conceptually, I think it's exactly like this. But the effect, whether the actual effect, we cannot be 100% sure. So also in terms of the number of years of this effect.
I did want to add to that and ask about, the whole hormesis thing, right, in terms of stresses. I know there was stuff about, alcohol, for instance, about it could be something like a hormetic stressor and then just the right amount. I know Sinclair used to talk about that with the resveratrol thing, and I'm wondering, what could explain why there's sort of a disparity of agreement on whether that has any tangible effect on longevity? Because, if you look online, I'm sure you've seen how many different people have opinions on this, and I wonder what efforts are being taken to make that understanding less ambiguous.
Well, hormesis may be understood as a mild stress.
Mm-hmm.
To me, it's a degree of stress. I assure you that severe stress increases biological age.
Mm-hmm.
Mild stress may be beneficial. There's a product who says
What doesn't kill you makes you stronger.
So it's because there are adaptive processes in the cells or in the organism, right? So you stress an organism responds by activating protective system. And actually we see this also when we compare old-lived species, long-lived species. So for example, we find when we compare, try to relate omics patterns, age of aging and long-lived species, we find they're slightly correlated. So basically this means that long-lived organisms like whales and humans, they are older, at the same kind of age than mice. But why would they be? But I think they're just better protected. So, and this same protection is achieved also, it happens during aging in long-lived species, and basically also may be related to hormesis, which is a mild stress. Another example, we have studied sleep deprivation.
Mm-hmm.
And there's a lot of data in the literature on sleep deprivation. They study it kind of negative effect. We look at our clocks, we actually find that sometimes there's no effect, sometimes even slight rejuvenation. Makes no sense.
I read that.
But if we do this like a longer sleep deprivation, like instead of just 1 day, like in mice, or 5 days, then we see increased biological age, which is also reversible.
It's really, this whole thing is sort of interesting because, then I also think of senescent cells, right? And this idea that they could potentially be harmful. They could, in a way, could that be like a self-induced stress that then sort of forces your body to respond to that hormetic, right? Are there certain mechanisms in either humans or other organisms that we can learn from where there's sort of self-induced stresses that act as a hormetic stress, a reminder of what the body should be going afterwards? Because it's like the idea if you don't use it, you lose it type of thing. And I wonder, are there ways of creating these very controlled damages in a body and getting it to sort of do that automatically, and it sort of keeps the thing going to a degree? Is that making... because senescent cells are a stress, sure?
Yeah, there's a fine line between mild stress and severe stress. We've been discussing these models. For example, maybe related, I think it may be related, for example, call it damage vaccine. Right? Can be actually vaccinate an organism by introducing certain types of damage. These are valid approaches, but experimentally not easy to design experiments. But potentially you may be right. It may be possible.
That would be cool. Sorry for running. Anybody else have any other questions?
Yeah.
Great talk. I have a lot of questions. I'm going to try to merge a few of them. The clocks you were using for the organ-specific, that was protein and serum and plasma from the UK Biobank, and consortium protein for let's say liver would be all ASD and then whatever others. It wasn't methylation, right?
It was levels of proteins, changes in
specific proteins that would be relevant to that organ type.
So we first look at the gene expression and we find which organ expresses which gene.
Okay.
And because we detect them in plasma, we infer that they are secreted from that organ and go to plasma. And if you focus just on these proteins in the plasma and develop a model, aging model for them, for these proteins, then we can predict biological age of that organ.
Yeah.
That's how it works, but it's all based on protein levels.
Yeah.
So,
Okay. My second question was going to be, you mentioned that these epigenetic clocks seem to change based on time of day, like they can fluctuate throughout the day, and then the proteins in your body can do that as well. And I'm thinking about the circadian element of, depending on the organ type, some of those may fluctuate more on time of day or might be diurnal, right? Like the thymus or any sort of hormonal changes that would go on throughout the day, those would have particular fluctuations. So I wonder, I mean, I'm sure that can easily be controlled for, but I wonder how much, which organs would be most plastic in that. And
there are no good data sets for it. We studied this at other levels, other modalities, but we don't have an answer yet. It's an ongoing project in the lab. But it's really puzzling. So, for example, you would think that after sleep, you're sleeping, you wake up, you should be, kind of youngest, right? We kind of remove damage that accumulated. But then if you look, for example, at the chance of dying, the highest chance of dying is in the afternoon, not in the evening, not at night. So we wake up, and then we live for a few hours, and then we at the peak of dying at that point. So why? It's and then also diseases, you look at the incidence of heart attack or stroke, and also the peak also at that stage.
yeah.
So,
But we have these fluctuations like ultradian cycles, right? So the afternoon sort of window, everyone colloquially knows just you feel sort of sleepy around that time. I don't know if that's exactly.
But it's the effect of food. I think after we might also age when we eat.
Sure.
When we immediately after we eat. Because
that makes sense.
I mean, I was trying to remember. Initially, I told you about the model. We always use that model. So in a simple way, we can view this as a level of damage in the simplest way. Yeah. And so, for example, under severe stress, like disease, this damage accumulates. Once the stress is relieved, damage can be exported. Yeah. And then we become younger. In the same way, we work hard on your project hackathon, you become older, and then you kind of sleep, and you become younger again. So it kind of fluctuates. But this idea of fluctuation in biological age, constant fluctuation, is not. It's a very new concept. I don't think. I think if we present it to the aging community, I don't think it will be easily accepted yet, because some people think of aging something which is only happens long term.
Sure.
But it's a very interesting kind of, we actually don't know how exactly to, last weekend I spent my whole weekend making selfies of myself every 15 minutes. And then we predict the age and how it changes during the day.
Your smiles slowly disappear.
No, I try be same a white background, same light, selfie constantly the whole day. Because there is no good data sets, but this is just one way to do it.
So you said that when we're exposed to stress, we age, and then when we relax, we eat, or when we get ourselves out of that stress, we go back to our age. So does it fluctuate and it gets back to the same baseline, or is it keeps on increasing?
The resolution of the clocks is not good enough for us to tell whether the organ goes back exactly to the same baseline or not. I often get these questions when I discuss pregnancy, for example, because pregnancy it's an increased metabolic stress, on the woman's body, and actually this also can be observed, gestational diabetes, incidence of heart disease. They all correlate. Third trimester woman is older, but then I'm asked sometimes, but we have a fetus, right? So there's also kind of, in effect, it's a parabiosis. So maybe a mother could benefit from a fetus because fetus is young.
Or maybe the fetus is taking all of the vitamins, all of the essentials from that's why she ages, and then when it's out of her body.
It may be. We don't know. But again, I think we need better data sets to quantify. We are not really sure. We know it approximately goes back to normal, but maybe it's a little bit high, a little bit lower. We don't know.
And how do you think you can measure that?
Well, with larger data sets, for pregnancy, we had mice. We collect blood. We make pregnant and collect blood before and during pregnancy up to postpartum, In humans, we go with data sets which are available, but it's typically smaller data sets because we need longitudinal. We need like blood from same women, like before they are pregnant and then during pregnancy. Typically data sets like 20 women, 50 women, just not enough. We need like 1,000 at least.
I have two questions. So when it comes to muscle breaking, I want to understand like how does it impact the biological clock?
You can hear me again?
Like when you're working out,
Working out? Mhm.
right? There's a lot of muscle breaking, right? So how does it impact the biological clock?
We haven't studied this. We don't know. We know the effect of exercise and generally positive in longer term, but how exactly, like during exercise, it changes, we don't know.
Post
Yeah.
exercise, like I want to understand like the biological clock of the muscle before exercise and post exercise.
And after, yeah, we haven't studied this, so we don't know. But I think it's possible. I'm sure there are data sets out there that can be studied with existing clocks.
And one more question I had is the temperature of the organ. So like this is like
Very
this
good
is,
point, yeah.
like the temperature. So this is like very intuitive understanding for me, but it's like I'm trying to like understand like if the temperature of the organ is warmer, suppose the heart has a warmer temperature than your gut, right? Is it possible to say that the heart, I don't know, is older in biological clock than the gut?
So I would think at that level, there's probably an adjustment in the rates, right? So organs develop is, not at the same rate. So for example, like neurons, they develop earlier, but the reproductive system develops later, and then but also ages faster. That's why I think we have this dramatic change in fertility, like menopause and so on, that we observe. But then we more think in terms of the differences in the organ temperatures over time. Like, for example, in response to some stresses or environmental conditions or historical periods. For example, like one of the ideas that we explore in the lab, like now people live longer, right? And when you visually look at people like 70-year-old people, they look like previously 60-year-old people. They look young. But when you look at younger people, it's opposite. So meaning that, for example, if you look at the age of puberty, like menarche over the last 100 years decreased maybe 3 to 4 years, and also in boys as well. And if you look at like age of teeth replacement or bone growth, it's all shifted to the young side. It seems like we're developing faster, but living longer. Why? It can be identified factors and one factor I think is temperature. So if you look at like 100 years ago, look at temperatures, the normal temperature was 37 degrees. Now it's 36.6. We decreased the temperature. So maybe this could be one factor. But this is a hypothesis, right? So I think temperature is very critical. We actually have a project on temperature in the lab. We expose mice to different temperatures and then see how they perform later. And we also go to the hospital, when children are prematurely born, they are placed in incubators. And for some reason, at different temperatures. It depends on the nurse. Some nurses prefer that temperature, and we can follow them. We're currently doing that.
I feel like whenever the body reaches a warm state, like Bruce, like this is so intuitive. Like I'm sorry, I like this smoking or drinking, the body gets warmer. Do you think like using warm temperatures, my own body is breaking it down? I mean, this is just like
Kind of in the long term, I think being warm it would be bad, right? So because they accelerate all the processes kinetically. But at the same time, becoming warm also activate protective system, adaptive systems. So at least initially, I'm not sure whether it would be necessarily. I don't think it might necessarily be deleterious attack. So yeah, I think so. In some countries, they may age with slightly different rates currently, but based on many factors, right? Genetic factor, could be environmental factors, could be just a rate of infections, like sanitary conditions make them get infected all the time. They have higher temperature, and it just happens many times, and therefore they age faster. Even then,
so every time you get sick, do you age more?
we don't know. I mean, this all is very, very new, right? So this biomarker, the first biomarker was developed 10 years ago. And we're still debating which biomarker like to use, how to construct, and we applied some levels, but we're just beginning here. Like I encourage you all like to join us, and you can apply all these biomarkers, develop better biomarkers to address all these questions. Most of the questions that like it's all very good questions, just nobody has done it yet.
All right.
My question is, where did the first born? It seemed like you talked about the enzyme damage in the beginning. Why does this damage only manifest itself at older age, like from the early age, from your baby to I don't know, 25, 30, whatever? You seem to be getting stronger and more powerful and bigger also, but then all of a sudden you start to decline. What causes this decline to start happening all of a sudden?
But it's not a sudden process of decline, it's a continuous process. For example, if you look at incidence of diseases, so mortality, like some integrating parameters like that, they just consistently go. So just damage accumulates over time, and eventually it results in manifestations that we see, right? Functional decline, diseases, and death, whatever. I think more interesting is what happens during development, because there is really puzzling patterns actually. So for example, newborn children, they're equally likely to die as 60-year-old person. It's quite high mortality rate initially, and then drops immediately. And the minimal mortality in humans, can you predict? Can anybody guess?
30?
30, minimal mortality? 9, at 9 years old. And it's like, it's kind of puzzling. Why 9? It's just not related to anything, right? Not before puberty. So we think it's an overlap of two processes. One is aging that we observe later. Initially, mortality is very low because of aging, but initially mortality is high due to developmental processes. So it's initially high. Actually, it's highest even during pregnancy, right? Fetal development, embryonic, and it kind of drops, and then becomes minimal at 9 simply because it's just one process goes down and one goes up. There's nothing special about it in that sense.
One other quick thing to interject in terms of the way temperature and thermodynamics could affect, the material, like the actual physical material properties of an organ. Have we found if there's anything between if an organ is not in terms of getting some type of a damage or anything, but like, do bones age at a different rate in a very noticeable way compared to softer organs? Because I also think in terms of plasticity and adaptability, it's sort of interesting that, like, as far as I know, that every organism that I've understood, that I know, it seems that anything that acts like a brain or a nervous system is basically always soft and squishy. And skeletal systems are sort of always by definition somewhat harder. And I'm wondering why that pattern emerges and what that says about aging at all.
No, somehow within the organism there's a coordinated somewhat at some level coordination between organs in terms of aging. There are some outliers. I already mentioned reproductive system. Maybe that's the main. I think ages faster. Some cells, like for example, like in the muscle, somebody mentioned muscle. We find that muscle stem cells, they age much slower. So, but not all stem cells. Some other hematopoietic stem cells like age like with rate equal to organismal aging, but some stem cells age slower. Yeah.
What I meant to say was like the actual like material properties. Is there something about the hardness and sort of that underlying structure of an organ, a cell, or whatever that seems to have an effect on the rate at which it ages? Because in a sense, it's like if something is highly plastic, then it seems like it would be able to better adapt to things, right? But then also if something is harder and it's colder and things are more packed together in that way, sort of thermodynamically, is that also aging at a slower rate? And I think that's also part of like this whole like cryogenics, I think is what they call it, about, there are these ideas that if you slow down, if you freeze something enough to an optimal temperature, it's almost like it's experiencing from its perspective, from its cellular computational perspective, it's experiencing less time because it just has less to do, right? And then there's less chance of something screwing it up.
Perhaps.
Okay. All right.
actually could I ask something?
Go for it.
I think when you're talking about cryogenics, that actually doesn't that essentially mean you're going through less metabolic processes?
Yes.
Which means you're accumulating less damage.
Yeah.
So,
I think hibernating species. do
Yeah.
that, right.
yeah, exactly.
So winter is good for us.
If you stay outside.
Yeah, but this with biology is very complex. Like hibernation, we actually study hibernation. I visited a year ago. I was in Alaska at the Institute of Arctic Biology, and they study hibernation, Arctic ground squirrels, bears, and so on. And what is interesting is Arctic ground squirrels, they hibernate for like 9 months, but every like 3 weeks they wake up. Like their temperature goes initially during hibernation all the way to low, like to 0. They don't freeze, but they keep 0. And then every 3 weeks it goes to normal, like 37. Like for 1 day, it just goes back. And I asked them, like, why do you think they kind of warm up every 3 weeks? The answer, to sleep. So hibernation is very different from sleep and from torpor. So it's really just metabolic suppression. So in that sense, it does preserve, like slows down, but the damage accumulates and they still need like sleep to get rid of that damage.
That also reminds me, have you heard of this thing called cessation before? Cessation.
Cessation of what?
Well, it was actually something from cognitive science. There was this guy, Ruben Laukkonen, and he was studying these very advanced meditators. I promise you this is more grounded than it seems. That would practice these certain things for very, very long times. And it would seem that they were able to sort of slow down their metabolic rate to the point that it seems like they say their consciousness, quote unquote, reached cessation, and it was like ceased to be there, and they didn't experience things or whatever. And I think there's some sort of theorizing in that paper about this was some type of cognitive hibernation almost, and that perhaps the brain was actually aging differently because it was experiencing less.
Maybe I would say it's more related to torpor, not to hibernation. Torpor is a state that in humans cannot be normally achieved, but like in many species, it can be. Like, bats, for example, they experience it every day. Even in mice, it can be used in mice and some other species.
Thank you. Anybody have any other questions?
Thank you very much.
One more, all right.
Last question. So like I'm trying to like figure out like how like cellular automata or like fractal expansion.
Which expression?
Fractal expansion.
Mm-hmm.
I'm trying to like associate fractal expansion with electric potential. So my thing, what Louis also said, is like everybody shares the same knowledge. Every cell shares the same knowledge of the incident. I'm sorry for one. Instead of being asked one person.
Well, like you can ask Helena, she works with my data. I think it's in that direction.
All right, Vadim, thank you so much.
Yeah.